Thanks for coming, everybody. So I'm going to be talking about some of my dissertation work looking at expression variation in island snake venoms. So selection can produce local adaptation and phenotypic divergence, often through coevolutionary interactions. The reciprocal selection that drives coevolution, it's typically not ubiquitous, right? It's confined within specific communities, creating this geographic mosaic of coevolutionary hot and cold spots, as described by Thompson and has been described in the Thamnophis and Rokyta system in the western United States. So if we want to study coevolutionary interactions, we need to identify these hot spots. So islands have long been studied by evolutionary biologists because they provide these sort of natural experiments that have been set up and run for thousands of generations. But they're also exceptional systems for addressing predator-prey coevolution because of changes in prey availability due to reduced species diversity on islands, as well as their relative isolation and thus limited gene flow. So islands are great systems for studying coevolution, especially predator-prey interactions, but we still need a system, right? And snake venom is arguably one of the best systems to study coevolution because venom is, by definition, a coevolutionary trait. It only functions after it's injected into another organism, and this obviously makes coevolutionary interactions with both predators and prey extremely likely. And selection as a result of coevolutionary interactions can produce adaptive phenotypic divergence, right? But if we want to understand adaptation, first we need to understand the molecular basis of the phenotype that we're interested in. And fortunately, the specialization of the venom gland towards toxin production and the fact that venom is a secretion, this allows us to use a joint transcriptomic and proteomic approach to identify the molecular basis of our phenotype, establish this link between genotype and phenotype, and thus link expression variation at specific loci to adaptive phenotypic divergence. So to determine whether snake venoms exhibit variation as predicted for a coevolutionary trait, I collected venom samples for 3 species of venomous snakes native to southeastern United States on both mainland and adjacent island sites. So I looked at the pygmy rattlesnake here on the top left, the cottonmouth on the bottom left, and the eastern diamondback rattlesnake. So my mainland population was the Apalachicola National Forest. It's in the panhandle of Florida. It's located just west of Tallahassee, seen in the inset up here. And my island populations were 3 adjacent islands that sit at the mouth of the Apalachicola River Delta. It was St. Vincent Island, Little St. George Island, and St. George Island. So these islands are Holocene islands. They're typically between 4,000 and 5,000 years old, and they vary in distance from the mainland. So St. Vincent is actually only a couple hundred yards from the mainland, whereas Little St. George is about 7 kilometers. And so I collected 12 mainland cottonmouths, 10 island cottonmouths, 18 island pygmy rattlesnakes, 10 mainland pygmy rattlesnakes, and 8 mainland eastern diamondbacks, and 11 island eastern diamondback rattlesnakes. And I restricted all my analyses to adult individuals to avoid the potentially confounding effects of ontology. So what we do is we collect the venom sample, we use reverse phase high-performance liquid chromatography to separate out that venom into its constituent components, and then we perform mass spectrometry of all of those peaks. And then we search our results against our species-specific transcriptome. That's how we create this GP map to establish this direct link from genotype to phenotype. We did this for each of these 3 species, and we found anywhere between 22 to 52 individual proteins that belong to 8 to 10 different families present in each of these venoms. So we use our armored approach to create this genotype-phenotype map. Well, how can we now use that to look at expression variation? So again, we use high-performance liquid chromatography, and you create this chromatogram that you see here. So using our joint approach, we can actually identify all the specific loci that produce the specific proteins that are present in each one of these peaks. And then we can actually quantify the area underneath each peak, and we can use that as an estimate of that protein's abundance and thus a proxy for expression. So for instance, if we're interested in this peak 2 here, using our omic approach, we can identify that contains a single protein, myotoxin. We identify the gene, we identify the protein product, then we can look and see how expression varies among populations for this specific locus. Now, unfortunately, when we quantify, we only get abundances in percentages, right? We only get relative abundances only in comparison to one another, and this generates a suite of statistical issues, okay? Percents, they're not dependent, they're only relative abundances, so we use log ratio transforms, which then allows us to apply standard multivariate statistical approaches to analyze our data. So we did that, then we performed a non-parametric MANOVA on each species comparing island populations to our mainland population. And we found that venom was significantly different for both pygmy rattlesnakes and the eastern diamondback rattlesnake, but not in cottonmouths. And as you can see, our p-value here is extremely high. And this is the first time really anyone has used this statistical model. So first I wanted to check and verify that cottonmouth venom really wasn't different between island and mainland. So what you're seeing here is a log ratio transform of the mean abundance of each HPLC peak. Okay, so you're essentially getting mean expression profiles for mainland populations in white and island populations in black for the cottonmouth. And as you can see, each peak is expressed at nearly an identical level. Right? So there doesn't seem to be any variation, any expression variation between island and mainland cottonmouth populations. But we did find that pygmies and eastern diamondbacks were different. So how are they different? So here's the same plot for pygmy rattlesnakes. As you can see, there's much more extensive variation here than there was in the cottonmouth, particularly at 3 different peaks. So peaks 7A, peaks 11, and peaks 33. So these peaks correspond to 9 different loci that are differentially expressed between these populations. And it seems that island animals are downregulating a single phospholipase, this phospholipase 3 here, while they're upregulating 8 different loci. So then we can look at the same thing for the eastern diamondback rattlesnake. And again, you can see there's much more variation here. And typically, again, at 3 peaks, peaks 1B, 20B, and 21, these correspond to 7 loci. And it seems that island individuals are upregulating a single locus, this metalloproteinase 1 here, while they are downregulating all these other loci. And what's interesting is that this is actually a pattern that we have seen before in the eastern diamondback rattlesnake. So the top plot is exactly what I just showed you. It compares island animals in black to mainland animals in white. Now the bottom plot is comparing adult diamondbacks from across their range in white to juvenile diamondbacks in black. And as you can see, the pattern is nearly identical. The same locus that is upregulated in island adults is also upregulated in island juveniles. And the same loci that are downregulated in island adults are also downregulated in juveniles. Putting the question, is this an example of pedomorphic dwarfism? Or is it the fact that I just can't tell a juvenile from an adult? Right? That's a good possibility. So Waldron et al. In 2013 found that eastern diamondbacks reach sexual maturity at about 1 meter in length. And we find that the ontogenetic shift in venom composition that this species shows is very tightly correlated with that. And once these animals reach 1 meter in length, their venom switches from the juvenile phenotype to the adult phenotype. So in all my analyses, I only used animals that were over 1 meter in length. Additionally, 7 of the 11 island animals I collected were gravid females. So these are obviously sexually mature individuals. So I just did a simple t-test, compared snout vent length as proxy for size of island animals to mainland animals, and found that island animals were significantly smaller on average than mainland individuals. So it seems that not only are island diamondbacks retaining that juvenile venom type, but they are also staying much, much smaller. And rodent trapping on these localities, it shows that, so the hispid cotton rat, Sigmonot hispidus, is very abundant on these island sites. But that's pretty much the only available prey source. And on the mainland, the prey assemblage is much more diverse. You have things like neotoma, rabbits, much larger prey items. So it seems that maybe this pedomorphic dwarfism is adaptive in this resource-poor environment with only very small prey items. Okay, so we identified that both pygmy rattlesnakes and eastern diamondback rattlesnakes have different venoms on mainland and island. We identified the specific loci that are responsible for those differences. But are there any trends? Is there anything common that's shared among these? Well, proteins expressed at low levels are often predicted to exhibit more variation than proteins expressed at high levels. You think proteins expressed at high levels are probably important, either in some pathway, performing some function. They typically evolve under strong purifying selection, and their expression levels are relatively conserved. So if we apply that to our system, you would think abundant toxins, they're probably good at subduing and incapacitating prey, no matter what type of prey it is. So maybe they're going to be retained, and all the adaptive variation that we're seeing would be restricted to proteins expressed at very low levels. So what I did is I separated our data set into a high expression data set and a low expression data set, and I reran our statistical model. And we found that for both pygmy rattlesnakes and eastern diamondback rattlesnakes, all the variation that we're seeing between populations is confined to the proteins that are expressed at very low levels. That the highly expressed things are conserved between these populations, and that all the variation is confined to things expressed at low levels. So what we find? Well, in conclusion, so 2 of the 3 venoms demonstrated phenotypic variation as predicted for a coevolutionary trait. The only one that didn't was the cottonmouth, The codmouth, it's a semi-aquatic species. It may be swimming back and forth fairly frequently, so maybe gene flow is swamping local adaptation. It also has a fairly eclectic diet, making coevolutionary interactions with specific prey species very unlikely. We found that diet-specific variation may be causing the variation on the locally expressed loci that we're seeing in the pygmy rattlesnake, and both the venom and morphological differences that we saw in the eastern diamondback suggest that maybe this population is exhibiting pedomorphic dwarfism. But I think most importantly is that our omic approaches, they allow us to identify not protein types or protein families, but actually specific loci that are differentially expressed between these populations, and that it seems that at least over short time scales, again, these islands are only 4,000 to 5,000 years old, over very short time scales, it appears that adaptation is happening in things expressed at relatively low levels. So very quickly, I thank some people for help in getting to sites and collecting samples, and these three agencies for funding, and I have a couple of minutes for questions. Yeah.
How do you know the age of the islands? Those things seem to appear and disappear fairly quickly, don't they?
Yeah, so they've done some core analyses, some geologists at UF and Florida State have dug some cores into the center of the islands, and they found that the oldest sediments there, they date the island to be about 4,000 or 5,000 years old. It's no older than that. I mean, their oldest estimate, I think, was 4,800. Yep.
Was there a significant difference between things that you categorized as expressed high and low? Like, is it, you know, where did you draw that line?
So all I did was I took a log ratio mean of each peak, and then I just separated out as evenly as I could. So there's 24 peaks, I took the 12 highest, made them one data set. The 12 lowest, that was another data set. Then I analyzed those independently. And all the significant variation was confined to things expressed at low levels.
You said that the established distinction between juvenile and adult is the 1 meter length, but that the island populations are generally smaller. So do you have any sense that that's still the cutoff point on the island, or is that?
Yeah, it seems that, I mean, so that cutoff point is still at a meter, but it seems they're not getting much bigger than that. So the biggest individual I've seen on this island is just under 4 feet, and that's very small for an eastern diamondback rattlesnake. So, I mean, there's still, so the smallest gravid female that we've seen out there is about 96 centimeters. I mean, and the 1 meter, it's not a very hard cutoff, right? I mean, there's probably plus or minus 5 centimeters in either direction. But no, it still seems that they're becoming sexually mature at a meter in length, but they're not getting much larger than that. Yeah.
So I know you said the islands are 4,000 to 5,000 years old. Do you know anything about the recruitment time for the snakes and or the prey?
Can you repeat that? For what time?
The recruitment time. So have the snakes been on the island since they separated, or were they?
So, I mean, we're in the process of analyzing rad-seq data, right? So that'll give us a better idea of that. So we've done Site D and ND5 for both the mainland and island populations, and there's no difference at all. So either it's extremely recent and there hasn't been enough time for divergence to occur, or there's still high gene flow back and forth. Yep.
You put out some hypotheses for why the venoms that are in the diamondbacks based on the system diet. But what about the pygmy rattlesnakes? Does it break some of those?
Yeah, I mean, the pygmies is tougher, so the island they're present on is very similar to the main. So the island that all the diamondbacks are coming from is very, I mean, it has one little hole, permanent body of fresh water. It's a very harsh environment. You know, the pygmies, the island they're on is very similar to the mainland. There's a lot of habitat heterogeneity. I mean, there's, I think, like 6 different species of frog out there. There's lots of lizards. It's pretty much everything you see on the mainland herb-wise. The diversity is the same on this island. Now, abundances and things like that. Yeah, there's no obvious dietary difference there. Yeah.
Very good between that close island and the farther apart island, particularly between the rattlesnakes and not just compounds.
Yeah, so the pygmy rattlesnakes are actually only present on the close island. They're not present on the further away island. And the diamondbacks are they're on that close island, but they're not very common. But that's one of the things I want to collect more samples, do rad-seq data, look to see if that's acting as a stepping stone for them to get out there. Or is that actually a mainland population of what's going on there? Yeah.
You said the differences were in those differentially expressed genes that were expressed at very low levels. Is it still possible that those might be particularly costly proteins to make, at least in the case of the down-regulated ones?
Yeah, I mean, every protein is costly, especially
but maybe
they're producing so much. But yeah, no, I mean, just because they're lowly expressed doesn't mean they're not.
we're going to have to call it there.